Everybody this morning. I think most of you know me. Obviously, I'm not Dr. Chen. Carrie Earthman. And, whoops, let's see if I can get this up and running here. I have the pleasure of talking with you about amino acid and protein metabolism for the next 3 class periods. And call me crazy, but I always invite myself to do this because I used to teach amino acid metabolism and vitamins and minerals back at Virginia Tech at my former life. And I like amino acid metabolism. So how many of you, you don't have to say this because I said I like it, but does anyone like biochemistry and amino acid metabolism? It's kind of, yay, all right. You know why I like it is because it has huge ramifications for clinical nutrition. And so those of you that know me may know me as your instructor for MNT. How many people are in MNT right now? Okay, most of you, right? So I'm, but some of you maybe I don't know yet. So, protein metabolism and lean tissue assessment is my area of research. It's something that as an ICU dietitian, I was acutely appreciative of even before I came back and got my PhD. And that's because lean tissue, skeletal muscle is a reservoir of amino acids for us when we are mounting an immune response or we're dealing with infection or trying to recover from major surgery. So as a dietitian, I was very appreciative of the role that lean tissue has in maintaining our health, right? So that's kind of where my orientation is. And amino acid metabolism is really important. It's critical. Those of you, how many are aiming for a career as a dietitian? First, dietitian? Okay. How about medicine? Fantastic. Nursing? Great. Ruth? What else? Physical therapy? Anybody? Dentistry? Are there any other health professions that I'm at? Pharmacy? Great. All right. So we're well represented. And then there are other people that are just living your lives, right? Which, of course, protein metabolism is critical to all of us just for that reason alone. So there we are. I got you. Whether or not wherever you land, hopefully you'll find this to be an interesting session. I'm going to start extremely basically. That's really not good English, but anyway, it's early 10:40. I know it's not that early, but I used to teach metabolism at Virginia Tech at 8 o'clock in the morning. So, if I get loud and boisterous, it's because I have a habit of that with amino acid metabolism. But I'm going to start really basic, and, forgive me for saying that if you've forgotten some things, okay? So that said, there are no dumb questions in this class. Marie? Wow. Well, the first thing you're supposed to do is turn around as an instructor and make sure that you have the screen down and that you're... Is it doing it? Yeah. Thanks for, pointing that out. So, we're not, how many years have I been doing this and I forgot to look behind me. Sometimes, you'll be looking at the screen over here and it's not projecting up there at all, and then you know. Huh? It does make it, the idea is really to keep you awake, make sure that you're paying attention. Okay. All right. Thank you, Marie. All right. So now let's start. You remember the structure of proteins? And if you ever looked at pictures of proteins, they're beautiful. Every protein has a unique structure, and it's really cool looking if you look at it on a 3D electron microscope. Okay, we ready? So, the general rule of thumb for the protein structure, amino acids joined together by peptide bonds. We've got basic structures, and you remember these maybe from biochem, primary, secondary, tertiary, and quaternary. We'll talk about each of those so that we're totally clear. There's a website there that if you want to, you can check it out. And I'm not sure if I took this out of your slides, because I did, didn't I? I think I did because, I'm sorry, I probably, let's see, why did I do that? I'm going to, because I'm not connected now to the internet, I'm not going to show you this. I'll tell you what, I'll repost this so that I'll check it first of all, because I think last year I checked it and it was a little bit hard to see on the big screen. It's one of those things that when you pull it up on your personal computer, it's really small, but if you look at it, you can kind of get the feel for, you know, the structure of a protein. So I'll take care of that. I'm sorry about that. I forgot I removed it from your notes. Okay, you've seen the structure of an amino acid before, right? So you know you've got this alpha carbon, and so we call this the alpha carboxyl group and the alpha amino group, and then we got an R group up there. We'll talk about that more in a moment. Are there any questions? I'm hearing some talking, and it's a room that's really hard to hear if you're in the back, and my voice is not so strong. So if you have any questions, please feel free to raise your hand, and I'll be glad to answer. Thank you. All right, so when we talk about amino acids joining together with peptide bonds, you remember how that happens? It's a dehydration reaction, right? So we're going to lose water and form a peptide bond, and I'm going to tell you this is in red on blue. That breaks all cardinal rules of PowerPoint. You should never put red on blue because it's really hard to see, so it's just how I ended up getting that shown there, so hopefully you can see that. That's peptide bond. And now, what do we call this? We have two amino acids together. What do you call that? What's it called? A dipeptide, right? So two amino acid residues makes a dipeptide. This is a summary of the very basic summary of the structures, right? So you remember primary sequence is just the line of amino acids connected by peptide bonds. Secondary structure is maintained through hydrogen bonding, and you get the two options there are alpha helix, where the hydrogens are interacting with the oxygens on the other on each of the residues, or we get beta pleated sheets, and those are secondary structure. Tertiary is interaction as the protein will fold up and they start to bring amino acid residues close together. What kind of interactions are associated with tertiary structure? Anybody think of any? Yes? Okay, so let's not, we're going to talk about R groups, but it's kind of at the level of what kind of interactions are happening between those R groups, potentially. Anybody? What kind of interactions are we talking about? Yes? Okay, right. Oh yeah, they're right up there, aren't there? But there's some other ones too. I didn't say all of them. Sorry, I forgot. Again, looking at the slide. It's been a year. Ionic interactions, disulfide bridges, what else? Can help to, what kind of amino acid residues tend to end up in the middle of a protein structure? Hydrophobic interactions, right? So you guys are right on par, you're remembering. There are also things that are very weak electrostatic interactions called van der Waals forces, right? However you spell walls, van der, D-E-R, W-A-A-L-S, right? Electrostatic. So. And then we've got quaternary structure, which is when you got two or more polypeptide chains, and those are also driven by some of the same types of interactions that we just talked about for 3D structure. Do all proteins have all levels of structure? No, right? Which ones are missing from some proteins? Quaternary. I suppose it's possible also that the tertiary might be missing if they're very small proteins. But usually you got at least primary and secondary. All right, so that's primary structure. This is, what would you call this one? What is it? What is this? A tripeptide, right? Because it's got three. We'll just go quickly. This is a beta pleated sheet, again. And these websites I checked out last year. I probably needed to have done that this year, so I will double check all of these when I get back to my office. I'm sorry I lost track of doing that. But you've seen them probably in your biochem textbooks as well. And then we got tertiary structure, and the idea here is that they may be far away from each other in the primary sequence, but they come together, right, and they help stabilize the structure. Incidentally, just out of curiosity, right, why is it that the hydrophobic ones end up in the center of a protein? Right, because they're in our blood, right? Now we're talking human, so we're gonna stick with the human body, right? Proteins are in an aqueous environment because we are aqueous creatures on the inside. So hydrophobic will be on the inside for the most part. And obviously on the other side of that, hydrophilic would be on the outside, right? All right, and then quaternary structure, and this is an example of hemoglobin. Hemoglobin has 2 beta chains and 2 alpha chains, and they come together to make a globular kind of protein structure. We'll talk more about hemoglobin in a moment. Okay, so here's our roadmap. I'm going to try to bring us back to this as we transition into each of the next topics. So that was pretty basic, I realize, so I appreciate your patience in that. Also staying fairly basic, I want to repeat or just kind of reiterate some of the key points about protein digestion next. And watch it, this is such an exciting animation. Should I go back and let you see it again? It's anyway, so we're going to stay here right now and talk about protein digestion. Ultimately, we're going to end up in talking about amino acid metabolism and each of kind of the big organs that we need to make sure that you are aware of in terms of metabolism. So first of all, let's talk about protein sources, right? Where are we getting protein presented to the GI tract, right? When I say GI tract, those of you that are clinical probably are aware, gastrointestinal is what I mean, right? So the GI tract where we're digesting and absorbing protein from. So first of all, exogenous, right? That's coming from our diet. If we're in a hospital ICU, we might be getting it through intravenous or a tube feeding, right? If we're not able to eat, but otherwise we're going to be getting that through diet. And I'm just representing this with a value so that we can kind of look at the proportionality of where we're getting protein from. Look how much comes from endogenous sources, right? Endogenous sources in the GI tract itself. So you remember how frequently cells in the GI tract turn over, right? About every 24 hours or so we replace all of the cells in our GI tract. So that's sloughed cells. We've got a lot of digestive secretions. What else? What other sources of protein are coming into the GI tract from endogenous sources? When I say secretions, what's in that? What kind of stuff is in? Where is protein coming from in those secretions from the GI tract? Anybody? Enzymes, right. A lot of them are going to be enzymes. Mucus is also a protein. So we got a fair amount, right? About 70 grams to the 100 coming in. That's the proportion. So a lot of it's coming endogenously. And all of that is coming into the GI tract. And look at this. How much is going into the fecal route of excretion? Very little. So we're very efficient. That's the number one point about this. We're really efficient at our conservation of protein. 170 grams coming in, only about 10 going out. And that's in the normal, right? In the clinical setting, you might have a lot of things interfering with that, some disease state that would impair absorption. But we are very efficient at absorption, so that's kind of our main point there. I'll just point this out here. This is a separate side point to this because we're really going to be talking about the GI tract next. But there is some nitrogen that also goes out in other forms, right? Integumental, coming from skin, hair, sweat. We'll lose a little bit through that kind of thing. And the other point that if I forget to say it, I hope I will remember to say it when we start talking about the liver. We're going to talk about urea and other things like that. But one of the things that happens in the colon is that we will recycle some nitrogen coming from urea. Urea ends up in the colon. We'll kind of reabsorb nitrogen at the colon. It's the only thing we do. We don't absorb amino acids from the colon, but some of that urea that gets made in the liver gets recycled in the colon. And we also get some nitrogen absorption from ammonia. And ammonia is, we have gut bacteria in the colon that'll break down urea and release ammonia. We're going to, hopefully by the end of this 3-day period, you will remember and appreciate how ammonia gets released from urea, which is made up of 2 ammonias, right? So, or at least 1 ammonia. I've forgotten the structure of it at the moment, but anyway. So no amino acids are absorbed in the colon, but we'll get some nitrogen coming in from urea and ammonia. Okay, now we're going to move on and just very briefly hit on protein digestion, which you probably had, what, was it advanced human where you might have had this, or maybe even the previous class. So hopefully this is a review for you. So protein digestion begins in the stomach, right? Protein hitting the stomach stimulates the gastric mucosa to release a very important hormone that stimulates 2 types of stomach cells to release HCl and pepsin. So does anyone remember, first of all, let's talk about the hormone. So just this, the gastric mucosa releases a hormone that makes you make or release acid, HCl, and pepsin, actually, in the form of pepsinogen. Anybody remember the hormone that makes you make acid? Gastrin, very good. Gastrin, right? Gastrin is released from the stomach mucosal cells. Anybody remember which type of cell? This is physiology now, I think, probably, maybe. Which ones release HCl? This is like trivia. Wait, can you say it louder? Whoever said it? Parietal? Is that parietal? P-A-R-I-E-T-A-L. Shall I say that again? P-A-R-I-E-T-A-L. Parietal. Those cells release HCl. Anybody remember what type of cell releases pepsinogen? Yay, fantastic. Hey, you guys, some physiology whizzes. Chief, C-H-I-E-F, chief, right? Okay. So gastrin acts on the parietal cells to release HCl. The chief cells release not pepsin, but pepsinogen. Pepsinogen is the precursor to pepsin, right? So, whoops, excuse me. What does the HCl do? Go ahead. I'm sorry, hands I'll say. Yeah, go ahead. All right, first thing, activates pepsinogen to pepsin, right? The active form of pepsin. So that's great. What else might it do? Yeah, lowers the pH. Why is that important? What does that do? Yeah. All right, it denatures some of the protein, right? What do we mean when we say denaturing? Breaking it down. Think about it in terms of structure, and we're going to talk more about this, right? It's going to, go ahead. Okay, so it's going to start disrupting some of the structures, right? Start to unfold the protein. Now, how does that help us? All right, this is down, right? Hopefully all of you are thinking these things, even though you may not be. I hear a lot of whispering, so I think you probably are. It's going to make those amino acid residues that are in the primary structure more available, right, to the pepsin to start acting on it. So enzymatic breakdown can begin in the stomach, right? And the acid is critical to make that happen. What else does the acid do for us? And this is not related to protein digestion. Go ahead, whoever, Ruth or... Yeah. Acidity, right, lowering the pH helps us against, when we're at the state fair and we're eating some food from a food stand that may or may not be completely 100%, hygienic. It doesn't always save us, does it? But it's kind of our first line defense, that lowering acid. So something to think about, right? Are you aware of, and I hope some of you may actually have to be on a medication for acid reflux. Anybody know what kind of a medication? It's one of the most popular one right now, overprescribed. You probably know someone that's on it if it's not yourself. What'd you say? Prilosec. What's Prilosec? It's an antacid. What does it do exactly? What class is it in? Anybody know? So it reduces acid output. It's a proton pump inhibitor. Very, very effective at shutting down acid production. So it's just something to think about. Keep it in mind, right? So people that are on that may very well be compromised a little bit, right? I mean, they're very effective. You typically take them every 24 hours, and people are on them for years. And we don't have a lot of data to talk about it and to look at it, but we've shown that there are some impacts of that. It's not the only way we digest protein. It's the beginning of it, right? So it could be that they may have some issues. There are other issues with B12 and other things like that, but we're not talking all about that right now. Okay, what does the, so what does the pepsin do? Obviously, I've got that on the picture here. So pepsin begins the process. It's kind of our first line enzyme. We're going to start getting some free amino acids, but mostly we're going to be chopping up those proteins and yielding large polypeptides, some oligopeptides, medium-sized things. What does HDL do to peptide bonds? Will you say? Whoever heard us. This room is terrible because it's... Anybody want to say? What does it do to peptide bonds? HCl. Nobody? Come on now. Breaks them? Show of hands. How many people agree? Does it break them? Say it louder, whoever's speaking. Doesn't do anything. You're right. It doesn't. Right? We need endopeptidases or exopeptidases. We need enzymatic cleavage. The peptide bond is extremely strong. So, HCl just gives us the setup, right, to help denature the protein, expose that primary sequence so we can start cleaving off amino acids, but it really doesn't do anything to the peptide bond. That's going to be on the exam, okay? I mean, so just make sure that you know that. Okay, protein digestion then is completed in the small intestine, right? We've got intestinal peptidases and pancreatic proteases. I'll show you on the next slide how that works, but they work together to attack peptide bonds. Okay, so they're the only things that are going to break those peptide bonds, or peptidases. We got exo and endopeptidases. Can anyone tell me what the difference is between an exo and an endopeptidase? I'm an amino acid sequence. This is the primary sequence. Where's the exopeptidase going to act? On my hands. One at a time from the outside. Endopeptidases tend to be specific for certain amino acid residues, and they'll chop them up in the middle. And we need both of them, right? Obviously, the exos, if we only had exopeptidases, we'd never get anywhere. So endopeptidases and exopeptidases working together. Do I need to repeat anything? By the way, and if I speak too quickly, please raise your hand and let me repeat, because I know sometimes I talk fast and it's not that early, but it's kind of early, so. Okay. All right. Now let's look at what happens in the interior, we're now in the lumen of the small intestine, right? So the first thing is up here, entry into the duodenum, the first segment of the small intestine, you may recall from physiology, right? Right after the pyloric sphincter, we've come out of the stomach, and we're now in the duodenum. And you got this acid chyme, which is essentially a mix of stuff coming from the proteins that we've just eaten, the acid, the pepsin, right? And so we've got this polypeptide and oligo and amino acid, di and tri, and small peptides, right? Mixed, hitting the duodenum. And what happens first is well, not necessarily first. This sort of happens simultaneously, actually. But I'm going to do it as if it's a linear process, which is what happens when we do paper biochemistry. So that hits the duodenal cells. The intestinal endocrine cells will then release two very important peptide hormones, CCK and secretin. And I'm just looking ahead to see whether I've spelled. I haven't. Let me just see. Okay, shall I spell out? Can you all look up cholecystokinin? Do you remember how to spell cholecystokinin? I'll spell it, okay? CHOLECystokinin. We could do a spelling bee, of cholecystokinin. CCK. You can remember it as CCK, okay? But if you see cholecystokinin, you should recognize that that's CCK, okay? Anyway, they're regulatory peptides. They get released by the endocrine cell, and they go. Let's first talk about what they do at the pancreatic cells, the pancreatic acinar cells. Very important impact on the pancreas. So that's why they're called hormones, right? Remember the definition of a hormone is that it's released by one cell or organ, and it travels through the bloodstream to another site, a distant site, and it acts there to have important function. So the pancreatic cells do two things. One is that they will release trypsinogen. They also release sodium bicarbonate, right? What does the bicarbonate do to the duodenum, the stuff in the duodenum? It neutralizes the acid, which means what exactly, if you had to explain that? What does that do? When we say neutralizing the acid, what is that, just biochemically speaking? Raises the pH, right? Just make sure you're remembering what those all mean, right? So you raise the pH. We'll talk about why that's important in a moment here. And then this other thing that CCK, and maybe I'll back up just a tad here. I guess it all comes at once. Okay, here we go. So back to this. The CCK and secretin are also stimulating the mucosal epithelial cells in the intestine. So they're acting distantly at the pancreas, and they're acting locally at the epithelial cells of the intestine to release this important enzyme called enteropeptidase. Okay? Look at what enteropeptidase is doing. Enteropeptidase is the key enzyme, excuse me. Key enzyme. Because it's activating trypsinogen to form trypsin. If you didn't have enteropeptidase, you would be significantly limited in your ability to digest protein. And you know why? Because trypsin, once trypsin is formed, it autocatalyzes the reaction to make more trypsin, so it acts more on other molecules of trypsinogen. And it also activates all the other pancreatic zymogens, those proenzymes that are produced by the pancreas. So you can see, the pancreas has a critical role, obviously, in protein digestion. I know you all remember that. But this enteropeptidase is key. Without it, you're really not going to be able to activate that first molecule of trypsinogen. And that's, again, all this is happening simultaneously. Once you get any trypsin around, you're going to autocatalyze, so it's great. But it is very critical, this enzyme. So if you have a genetic defect in enteropeptidase, which is extremely rare, but if it were to be the case, you would have difficulties with protein digestion. Any questions about any of that? Remember that? Yeah. And I should have a picture of this, but I think they are integrated, and I could try to confirm that and look at a picture to show you, but I think they are, because we're talking about, as food is moving through the duodenum, so I'm not sure what proportion they are. That's a great question, actually, but I'll try to get more clarity. If I can find a picture, I'll show you one next time. Any other questions? Okay. So you remember that these are called zymogens, right? Or the other way, and that's Z-Y-M-O-G-E-N, or proenzyme, right? Because they're in the inactive form as they're released by the pancreatic acinar cell. So next step, showing this again, got the stomach, we got pepsin and HCl forming these larger polypeptides, oligopeptides. Then we've just talked about pancreatic proteases and amino, di, and tripeptidases that get activated. Incidentally, we talked about the bicarbonate coming from the pancreas, and I forgot actually, well, maybe I will go back just one slide, sorry. What is the importance of this, right? We talked about neutralizing the acid. What do you think that does? Why is it important that we raise the pH in the duodenum? Sorry, Marie, you just...
It does not destroy that high acid or the low pH destroy that tissue inside.
Okay, let me, so that's one possibility. What were you going to say, Marie? Okay. So we've got, so the acid doesn't break down the intestine. So by raising the pH, inactivating the enzymes, activating them, right? So, you're all thinking about, okay, so if we had the pH of, 2.0 hitting the duodenum, surely there would be potentially some damage. So I'm not going to say that's totally off, but I want you to think about more appropriately, because what that's doing, it's neutralizing, right? So that's good. But the main reason that that's, in terms of protein digestion anyway, that it's so critical is that those pancreatic enzymes and incidentally some intestinal enzymes optimally function at a higher pH. And the pH in the duodenum is around 6. Okay? So we start to bring that pH that's in the stomach in many people, not people necessarily on proton pump inhibitors, but the pH is around 2 to 2.5 to 3 or so, in the most acidic state of the stomach. It doesn't, it fluctuates obviously. But then when we get to the duodenum, we're raising that pH to around pH 6, and it helps those enzymes, proenzymes, get active, right? They're optimally active. So that's all working together to help us better digest protein. So that's important too. So if you didn't get that, make sure you write that down. That will be on the test too. All of these are basic things you just have to kind of keep in mind, because you can then apply it. Okay, when you're in medicine and you're thinking about that person on proton pump inhibitors, you're thinking about, you can start to begin to put it together, right? Okay, let's go back to the small intestine lumen, right? We got the pancreatic proteases. We also have these, I'd called them intestinal peptidases. Specifically, they're amino, di, tripeptidases, other types of intestinal peptidases that are cleaving and acting on those smaller segments, and the pancreatic ones are breaking those bigger ones down. We have some small peptides that get absorbed across the brush border remember the brush border, very, very high surface area of the intestinal mucosa, that is so important, those finger-like projections. I know you remember that probably, right? Because that's so interesting in physiology. So we've got small peptides coming over, and we've got free amino acids. We'll talk about how that happens and what kind of transport that is in just a moment. And then we've got intracellular peptidases. So inside the mucosal cells, we have peptidases that are going to break down those small peptides into free amino acids. Oops. And I'll just say it here because I'm going to get back to it. We also have the ability to absorb across the basolateral membrane some small peptides. So some small peptides are somehow resistant to breakdown by these small peptidases. Okay, so some of them will get across. We'll talk a little bit more about that in a second. But a lot of them are going to go across as free amino acids. And just so that you have it written in a few places, the majority, actually, of the way we absorb amino acids is as small peptides. So more gets absorbed as small peptides than free amino acids. All right, so let's start up here. So active transport is the way this happens, that we get small peptides and free amino acids absorbed across the brush border of the small intestine. And so here are a few points I want you to write down. Number one, there are separate mechanisms, separate transporters for absorbing small peptides and amino acids, free amino acids. Okay, so there are separate transport systems for small peptides and amino acids. Point number two is the one I just said. The primary way we absorb amino acids is in the form of small peptides. So it's active transport at the brush border. It's passive, mostly, through diffusion at the basolateral membrane. Remember, the basolateral membrane is the surface of the cell that interfaces with the capillaries, right? So we absorb amino acids into the bloodstream. So for the most part, we're going to break down those small peptides that can get absorbed into free amino acids. And I said some small peptides remain intact inexplicably. It's not really well understood. Certain amino acid sequences and residues kind of, evade breakdown. And we consider those to be bioactive peptides. Many of them are bioactive, not all of them, but they have various activity. Yes? That is a great question. And I don't think I can answer that exactly. So charged amino acids do get across. And I'm not sure exactly how the mechanism works. It's been a while since I've thought about that. So let me see if I can look into that. You think about the structures, and there are at the brush border, it's easier. There are transporters. So that's a great question. Let me think about that. So by far and away, a lot of them are going to enter the bloodstream. Go through the portal vein from the intestine. The portal vein going directly into the liver, right? And we get a huge rise in amino acid concentrations after a meal. So that amino acid concentration increases several folds after a meal. And that's an important thing to just to think about. Some of the sources of I was talking about those bioactive peptides have been identified coming from whey protein and soy protein, and some have impact on blood pressure regulation. Some are thought to have some impact on appetite control and various things. It's a very active area of research. So that's an interesting thing that some can. And I have something in my notes that might get at your question, and I'm going to reaffirm it. There are some active transporters at the basolateral membrane. A good many of them are going to come through diffusion, but it could account for the ones that have charge. Let me see if I can confirm that. It's been a while. Okay, let's talk about amino acid transporters, just some basic principles. First is that they are redundant. And what does that mean? It means that one amino acid can be transported by a number of different transporters. Okay? Redundancy in transporters. So one amino acid will be recognized by a number of different transporters. We also have that competition is at play. So amino acids for the same carrier, right? So we might have a carrier that carries 2 or 3 or 4 particular amino acids. Those amino acids will compete for transport by that particular transporter. There's a lot of overlap. So the redundancy is one amino acid may be transported by a number of different transporters, but on the other hand, there's competition, so that one transporter might have 3 or 4 that are in competition for it at any particular moment. Okay? The other thing that's interesting is each tissue has a unique complement of transporters to meet its particular needs, right? So we have a functional system there. And then the last point is that transporter activity is affected by hormonal and physiologic state. And there's an example of this, is that glucagon increases the A transporter, which carries alanine, in liver cells. Why do you think that would be? Why would glucagon impact the transporter that carries alanine in liver cells? Yeah?
Alanine can be expended.
Yes, fantastic, right? So this is how we begin to pull metabolism and metabolic pathways together, right? Amino acid that's so important as a gluconeogenic precursor, right? Alanine, its transport is increased at the liver. The number of transporters for alanine increase in response to glucagon under conditions when we want to have more alanine taken up by the liver. So great. So things to just kind of think about, critically important changes. So a little bit more about amino acid absorption. Absorption characteristics of amino acids vary such that essential amino acids are absorbed faster than non-essential. Essential are absorbed faster than non-essential. Did you learn it as essential and non-essential, or dispensable and undispensable? It's confusing. Your book actually, I think, uses dispensable, and so dispensable means non-essential and indispensable means essential. I'll try not to mix them up. It's confusing. What else? Large amino acids are absorbed faster than small ones. That's just a couple of examples. And we have, again, the point that amino acids for the same carrier compete, even though there's some redundancy. So if you keep that in mind, and you think about specialized amino acid supplements that may be given for athletes, or athletes might take, maybe go to GNC and you get something that's high in, say, arginine and glutamine or something like that. What do you think about that? Just based on what we've just talked about, not whether you believe it works or not, but just biochemically speaking. What would you say to somebody who's thinking about supplementing, maybe taking a glutamine supplement or taking a glutamine arginine, leucine supplement, something like that? Yeah.
Just that sometimes the amino acids can have a deficiency in another one, using supplements.
And probably not a deficiency per se, just because you're eating other. Now, it's conceivable if you were only doing that, yeah, you could cause a deficiency. But again, how? Let's get further on that. So how, it may not be such a good thing. I'm getting that. That's the gist. Why? What would you tell someone based on what we just talked about? What do you think might happen if you You know, like I said, 2 or 3 amino acids, or maybe 4. Yeah. Okay. Yep. So that's kind of, you know, competitive inhibition can happen, right? You might swamp out the glutamine transporter and the other amino acids that might be brought in at a muscle, particularly skeletal muscle tissue. We'll talk about, we're not going to talk in that much detail about the types of transporters that are available at a grad level course. You might talk about, you know, individual tissue transport. Just in a general way, though, it could cause competitive inhibition, right? So that their goal was to beef up the intake of glutamine at the muscle, or arginine, or whatever, you know, leucine, probably more likely. And you might be causing an imbalance at that tissue because you're kind of swamping out, you know, the particular transporter. So, and then what else do we know about amino acid transport that you might steer someone away from taking an amino acid supplement? Free amino acids, right? Just amino acids. What else do we know? You look back a slide or two if you want to. And you can talk to your neighbor. Let's take 20 seconds if you don't have an answer, if no one raises their hands. I've got a hand. Okay, wait, you might be off the hook. Hey, right? So hopefully as you began your thought, you thought of anybody else want it, because I bet no one heard you at the front here. Anybody in the back want to say? I heard some talking. Why is it? I can repeat the question. So the question was, what else might you tell somebody? What else do you know about amino acid absorption that would make it not such a hot idea to supplement free amino acids? I got an answer up here, but I bet you in the back didn't hear it, so I'm going to give another chance for somebody up in the upper reaches up there, in the nosebleed section. What would you tell them? So you've told them that there might be some redundant, you know, competitiveness at the transporters, competition that would maybe swamp out. What else might you say? Are they going to absorb those amino acids as fast as any other form of amino acids? Yeah. Yeah, right. So are they doing themselves a disservice, really, taking a free amino acid supplement? And they're expensive, and they don't taste very good. I don't know, any of you mix up a free amino acid solution? It's important that you know what that tastes like so you don't start giving your patients free amino acid solutions without masking it with something, Rosie's lime juice, sherbet, you know, whatever. Free amino acid solutions are nasty. So, you know, you've just saved somebody a lot of money and pain, and that they don't need to be taking these. They really are probably detrimental to them. Any questions? I'm hearing some, yeah.
Can you repeat what the last answer was?
So the last answer was, sorry about that. This room is abysmal, isn't it? It's like a cave. I'm down here at the, hello, at the top. Okay, sorry. So let me repeat. The point is that you absorb small peptides faster and more efficiently than free amino acids, right? So, you know, you're really doing yourself a disservice taking an amino acid supplement because you're not absorbing those free amino acids as well as you would with just regular protein and you know, possibly a peptide formula, but you know, you don't really need to be doing that, because if you take protein in and you've got normal digestion, you're really in pretty good shape. You're going to create a pathway of multiple dipeptides, oligopeptides. You know, those oligos will get down to di and tripeptides. You're going to absorb those. You're going to absorb some of the amino acids too, but it's way better than doing an amino acid supplement. And the other part of it that I just said is that, you know, it's expensive and it's pretty much not a very useful thing to do. They can also cause some GI upset, right? So you get a stomach ache as a result of you know, taking these kind of things. So not a good idea. I've lost Jessica. When do you all take a break? Right now? Well, that was convenient, wasn't it? Well then, okay, let's take a break. You're not just saying that, right? I mean, you could, you know. Okay. We come back in 10 minutes. Is that you guys get a whole 10 minutes? I might make a hot cup of tea in the meantime. So see you in 10 minutes, 11:40. All right, I think we're going to get started again. I'm amazed how fast 10 minutes goes. And if you want, we can take a 30-second talk to your person next to you. I know it's a little hard to get in kind of groups here because we're linear, but if you have someone you want to talk to next to you, I've got a little case scenario, very, very simple case scenario. Person's admitted to the hospital, is on artificial ventilation, so is on a respirator, can't eat orally, essentially. Has a history of pancreatic insufficiency and has to be fed by a tube feeding. And you can pull all of your biochem together if you'd like and think about all the aspects, but at the very least, I want you to think about the protein aspects of this formula, all right? And think about what type of tube feeding formula would be best for this patient. So I'm going to give you 20, 30 seconds. Talk right now. Boop. What would you say? Thinking first and foremost about proteins. Okay, we ready? Is that long enough? Because I know what happens after about 30 seconds. You start talking about the weekend, maybe. Okay, all right, can we come back? Who wants to say? What would you say? What kind of formula would you recommend? And not a name, okay? Characteristics. What are the characteristics of the formula for protein? What would you do? It's kind of a no-brainer, so I think it's, maybe that's why you don't want to answer, right? What would you say? What are the characteristics? Can somebody say it louder? I heard somebody talking. Come on, you were just talking to each other. Were you really talking about the weekend? Yeah.
Just be a formula of small peptides.
Yeah, so small peptides and probably some amino acids too. So peptide-based is what we will say. So you've got some hydrolysis to create small peptides and some amino acids. And the reason for that, right, is that the pancreatic insufficiency means you're not going to have, and I'm not going to say it. Explain to me why. What's happening in pancreatic insufficiency to impact your protein metabolism such that you would need small peptides and free amino acids, okay? So explain to the patient. I'm the patient. Tell me why. Jonathan, you want to give it a go? Okay, can you put it more in lay terms? So he talked about we're not secreting as much trypsinogen. What else? What else is, yeah, it's hard to put it in lay terms, but this is, practicing the bedside manner. You gotta.
You're not able to break down some of the proteins.
Okay.
So giving you the proteins that you can absorb.
Okay. So I'm not going to be able to break down as much protein because your pancreas isn't working as well. Those pancreatic enzymes, you might raise it a little bit and say that. Is there anything else that goes on when we have pancreatic insufficiency that impacts protein digestion? Thinking ahead to the exam. What else does the pancreas do? Talk about pancreatic enzymes.
Secreting the sodium bicarbonate.
Bingo. Bicarbonate, right? You are not able to adequately neutralize that acid chyme to activate those pancreatic enzymes. Okay, this is critical. So both of those things are really important. And so we're going to do that. Now, we may also, the other things we can do is we can give lower fat, and that's fat metabolism. You guys probably have you done fat, lipid? Not yet. So that'll come later. At the end, we could make that a comprehensive question. And we might actually supplement pancreatic enzymes. We can give pancreatic enzymes to people as well to help them along, right? But the main thing we're going to do in terms of protein is give small peptides and free amino acid solutions. Those peptide-based formula are really helpful. Great, you guys are doing great. All right, summary, I'll let you read that later, but just maybe add out to the side another major point that I want you to take home is that which is absorbed better, small peptides or free amino acids, small peptides, right? And peptides are absorbed faster than amino acids as well. So it's the bulk of them are absorbed. More of the absorption happens as small peptides, and it's actually faster, in part probably because of the number of transporters as well. Okay, boy, we kind of hung on that. When do we end again? 12:35. All right, great. Thank you. You could have gotten away with, something different than that. Thank you for being honest, those of you that are. Darn. Okay, so let's talk about why do we need protein in our diet. This is also probably seems, maybe. Why do we need protein in our diet? What are you going to tell the person on the bus? Or the plane? I'm sure you've all had that experience, right? You're on an airplane and you say, you start talking to the person next to you and they say, what are you studying? And you say, nutrition, and then what do they ask? What should I eat? What do you think of the blood type diet? I love that question. I've started to say I work for NASA. Yeah, you get a little jaded after a while. I travel a lot, so I was like, no, not nutrition. Definitely not nutrition. Okay, what do you think about, whatever. Why do we need protein in our diet? What do we use protein for? Everything. Okay, that's the quick answer, whoever said that. What are we making out of, yeah. All right, building and maintaining muscle. So we've got structural proteins. What other types of proteins are we interested in building, right? Yeah. Immune proteins, we maintain our immune system. Okay, so we got structure, we got immune, so there are a whole lot of them, right? We just talked about a bunch in the GI tract that are critical, right? Enzymes are in every tissue. That's obviously not a complete list by any means. Another important class, though, that I want you to think about, and those of you that are in MNT, I'll give you a little leg up by saying albumin is one of them, very important one. What is the, there are a few functions of albumin, but. Okay, that's too in-depth, that's too MNT. You're right, but I'm not going to repeat that just because I don't want to confuse everyone, so I have to explain what that is, right? What does albumin do? It's a major protein in our blood. It's a carrier, right? It's a major transport protein. So it's carrying things like free fatty acids around, and certain drugs get carried. Medications will get carried on albumin. You know, calcium will bind to it. You know, we carry a lot of stuff. So carrier proteins, right? And we did learn about a bunch of negative acute phase, right? Retinol binding protein, you know, all these transport proteins. So a lot of different things. Albumin does other stuff too, but we'll leave it at that for this. So in order to build those, I'm just going to click because I know these are on your slides, right? We've got essential amino acids that we need to build those proteins, right? So we need protein as a source of amino acids, essential amino acids that we have to build other proteins, right? So we bring it in, break it down, use them. We also need a certain amount of kind of a non-specific requirement just to have nitrogen around, and that comes in also in protein. So besides protein, what else might we use nitrogen for? Is there any really essential, basic thing that we need nitrogen for in the body at the cellular level? Let's see how many hints I can give you. Building block of life, we might say. DNA, right? DNA is a nitrogenous compound. There are a lot of other ones too, actually, but DNA is critical, and RNA, right? We need nitrogen, so we need non-specifically, you know, a certain amount of nitrogen that's coming in. Some of it's coming in as amino acids. We're getting ammonia from foods. Nitrogen comes in different compounds, different ways. So we need that. That's why we need protein in our diet. And I've got these on the slide, so I'll click these. You know, why do we need protein, right? Kids, they're growing, they're building new tissues. Adults, we usually talk about maintenance. Under what conditions would an adult need more than just maintenance level protein? Pregnancy, right? Building a whole human being, is that the coolest thing ever, if you think about it? Right? There's a whole lot of protein. You're taking in a lot of nutrients as well to fuel that synthesis, but it's a huge anabolic state. What else? Injury, right? Those of you that are with me in M&T, we talk a lot about that. You need extra protein to help your body heal after injury. What else? What other physiologic state or condition might you need more than maintenance? Okay, we talked about pregnancy. Talked about wound healing. What else? Some of you in this room I know are doing it because you guys are young and yes.
Exercise.
Right. Exercise, weightlifting, weight training, right? Muscle building. Those are kind of the main ones. Incidentally, lactation as well, by the way, after pregnancy. Lactation meaning, you know, you're breastfeeding, right? So. All right. So what factors do we need to talk about when we think about protein and requirements and various things? So the first thing is nitrogen content varies protein to protein, but there's an average that we're going to use. Anybody know what the average percent nitrogen is? And those in M&T, I think you've seen this because you know. What is it? Not percent. Okay, so you've got the factor, Marie, in your head. Okay, so if you've got grams of protein and you divide by the factor 6.25, you'll get the grams of nitrogen. Anybody with fast math whiz could tell us what percent that is. Anybody remember the percent? 16, I'm hearing, right? That's an average, right? So the average protein is approximately 16% nitrogen. And I want you to write down the factor as well. I mean, you can do it. So if you got grams of protein, 80 grams of protein times 0.16, you're going to get a number for the grams of nitrogen. It's the same thing as dividing by 6.25, right? Am I remembering this number correctly? Somebody do the math just to play it out. Do you have a phone? Sorry? Okay, can you do it both ways? So if you multiplied it, 80, what was it, 80 times 0.16? Somebody else do 80 divided by 6.25. That's what you got. Okay, did anybody do it by 16? 0.16? Okay, somebody with a phone or a calculator. Get the same. Okay, great. So we're good. All right? So there are two ways of doing it. As a, certainly as a dietitian, that's going to be something that'll be in your head, how to convert grams of protein into grams of nitrogen. Essential amino acid content and proportions and digestibility and human variability. All of these things are important to consider. And we'll talk more about human variability and the factors in a moment. But essential amino acid content and proportions and the digestibility, those make up what we call protein quality. Okay, so the protein quality is the ability of a protein to be digested and absorbed, and the essential amino acid content and proportions. Now there are going to be some factors, human variability factors, that are going to affect the way we digest and absorb and various things. But protein quality, that's how we define it. And there are some other things that are talked about in your book around protein, net protein utilization, and various other things like that that come into play. But these are kind of the big ones. So when we think about amino acids, some have more nitrogen than others. Here are some structures for you. Did you memorize the structures when you were in biochem, or did they make you? Hey, so those are all right there, I bet, right? You can. I'm not going to ask you to draw them out this time, but I do want you to think about those structures because it's important for a lot of things in terms of, you know, the R groups and various things and how they differ. And you know, if we think about it, this right up here, this is an R group. You know, phenolic, right? Just if you think about that, what do you think? Is that going to be hydrophobic or hydrophilic? Did you learn that in biochem? You know, how they start? We're going to hit this again. We'll talk more about it. But what do you think? What would hydrophilic look like, would you guess? What kinds of attributes would you think might be likely to have it interact well with water? Anybody? Come on, biochem. Say it louder. Non-ring structure. Hey, well, yeah. This is hydrophobic. Because there are no hanging out, you know, you don't have the charges, right, that might interact with water. You think about electrostatic kind of interactions will make things hydrophilic. Anyway, we'll talk more about that in a moment. But focusing, some amino acids have more nitrogen than others, and this is glutamine, so it's got two, right? It's got one in its R chain. This one, each of these has two, asparagine, tryptophan. Wow, look at that one. How many's that got? 4, okay, 4 arginine, histidine, 3 lysine, 2 again. Incidentally, this one has the most. Arginine's got the most. 4 is the max, okay? So arginine is a very high nitrogen amino acid. So if you think about it, so a lot of our hospital-based formulas that get developed have a lot of arginine and they got a lot of glutamine. I just showed you glutamine has 2 nitrogens, arginine has 4. So guess what? The protein percent that you're going to see on the label of a formula like Impact glutamine has a lot of glutamine. It's also got arginine in there to promote wound healing, supposedly. It's going to be more like 18% or something like that. You can look on the label when you get out there in the world. So we said average is 16%, but they can vary depending on the content that is in that formula. Okay, 2 primary categories, and I know this is also, I hope, a review. High biologic value, right? HBV, high biologic value, and low biologic value. Complete or incomplete. I'm sorry, I'm clicking too fast. Let me just, I'm trying to get the titles here. So when we say complete proteins, typically it's all essential amino acids are there in the right proportion to support optimal protein synthesis. They are usually animal origin. The exception that I give you here is gelatin, because that's lacking in tryptophan. What other exceptions are there? Are they always animal proteins that are complete? Are there any? Soy, I'm hearing. Any others? Quinoa. Those are the ones I know, and there may be some other ones that are. So quinoa is a grain that has typically, thought to have complete profile of essential amino acids. Soy protein is a complete source of protein. So not all animal typically are going to be, for sure. But soy and quinoa are at least two examples. And if anyone looks it up and thinks of another example, bring it next time or shout it out. And the idea when we talk about complete essential amino acid profile, we're talking about the profile needed to support growth for 1 to 3-year-olds. That's the way the Institute of Medicine has reframed it. It used to be based on egg protein, and egg protein profile is pretty good too. It's a complete protein as well, but now the Institute of Medicine and the WHO point to the needs of a 1 to 3-year-old. So if we're meeting a 1 to 3-year-old's needs, we're meeting, we're providing a complete profile of amino acids. Low biologic or incomplete proteins lack 1 or more essential amino acids. There's a term, limiting amino acid is the essential amino acid that is in the lowest percentage in reference to that reference profile. Okay, so the limiting amino acid is the one that's in the lowest proportion compared to that reference that we're talking about. Usually they're plant proteins, again, remembering exception, quinoa and soy being two good examples of exceptions. And then what do we mean by complementary proteins? Anybody want to shout out what a complementary protein is, or that concept of complementary? Right. So you bring together two protein sources that may be lacking. They each have a limiting amino acid or more, but when you bring them together, it makes a full set, complement each other. Okay, well, that's on the next slide, so we'll go zip through that quickly and just want you to think about legumes are limiting in methionine, have lots of lysine. On the other hand, grains, lysine is limiting, but you got plenty of methionine. So one concept, and there are a lot of examples of this. So the idea is bring them together. They don't have to be at the same meal. As long as throughout the course of a day you tend to hit all of the different food groups, you're going to be able to do pretty well. We've got the concepts that I know you're aware of. The vegetarian. There are various forms of vegetarian. The most common probably lacto-ovo would be the ones that are they're consuming eggs and milk but no other animal. And vegan can range in how people interpret that. It can be a whole lifestyle in terms of clothing that is also avoiding any animal products as well. But when we're talking about diet, typically we mean no animal products at all. And plant proteins. Still, I think this is fairly true that make up 65% of the per capita worldwide protein intake, and around 32% of the U.S. Little bits and pieces I like to just bring up here, and that is the idea that corn as maybe you guys can remember this, maybe this is an example that you've heard about. So cultures that rely on corn as a staple grain. Any knowledge about that? What do you know about that? What comes to mind? It doesn't have to be anything that comes to mind. What's the problem with corn as a source of protein? Is it a complete protein? No, right? Do you remember what it's lacking or what the issue is with corn? Say it loud. Who is speaking? Sorry? Bound niacin, right. So it's got niacin in there. It's lacking an amino acid that is a precursor to niacin. What essential amino acid? Anybody know this? Is this a bit of a trivial pursuit question that we could make for nutrition? Tryptophan? Okay, so it's lacking tryptophan. That's the amino acid that's missing. So that's one issue. And then it also has a form of niacin that's bound up, right? What do we do to corn to help it When we're making, in cultures that really rely on that, it's been a huge plus. What's the deficiency syndrome related to niacin deficiency? Anybody remember? Pellagra, right? Pellagra, however you want to say it. I say it pellagra. You say pellagra. I say, okay. Right, so if you've got corn that has an unavailable form of niacin in it and it's lacking tryptophan, you're in kind of a trouble if that's your staple. So now what do we do to corn to help it along? Add lime. We treat it with lime, and that breaks down the niacin, right? Still an issue. So how do cultures that eat a lot of corn, say in Mexico where they're eating a lot of corn tortillas, or other South American types of countries where they eat a lot of corn, what do they eat with it typically? Beans, right? So we manage that by giving a source that has good tryptophan content. And if we can also treat the corn with lime and break down that niacin, then we've solved the problem of pellagra, which is really a devastating deficiency disease. So that's how vitamins and minerals come together. Kind of interesting, huh? Yeah, ready for the Trivial Pursuit game that I'm going to make in my retirement? Or you guys could help me now, that would be good. Oh, that is so blurry for me. That's really blurry, and I'm sure that's blurry for you. So I'm just going to point out that what's underlined here is higher biologic value and net protein utilization, which is in your book, and I'm not going to ask you about that. But just know that these concepts show up. Companies use them. They talk about protein content, and I don't think it says here. It doesn't say what % protein it is. But, eases digestibility. They're talking about digestibility. They're talking about biologic value, that kind of thing. So these things are important notions for you to think about. What else in terms of protein digestibility? Gee, where is that? Okay, a few tenets that we will think about. Animal proteins are better digested typically than plant proteins. And the second point would answer that question, why? Why is that? Plant proteins have some factors in them that kind of interfere with digestibility. So one of the things that plants might have are trypsin inhibitors present in some plants. Anybody think of a plant that has a trypsin inhibitor? Not sure where you might have learned that in your travels in this department. Soybeans. Okay, soybeans are one example of a plant that has trypsin inhibitors. Trypsin inhibitors in the soy are inactivated by heating. You remember what trypsin does, right? What does trypsin do? Quick. Okay, everybody's talking at once, but let me let you answer. Oh no, somebody else. Sorry, thank you though, Lane. Doesn't do anything to base. That's bicarbonate, right? Trypsin. What is trypsin? It's an enzyme, right? And it activates by auto-catalyzing other trypsinogen and also all of those pancreatic zymogens. Absolutely. So it's critical to protein digestion. Trypsin, very important enzyme. Strange, isn't it? I mean, I can't explain why, really. Soybean has trypsin inhibitors, but it does. So, not that you'd be tempted to eat raw soybeans, but wouldn't recommend it, right? So don't steam the soybeans. Eat the edamame as we if you go to a Japanese restaurant. Don't eat the raw. What else? Fiber, that also interferes, and phytates. Phytates are very high in soybeans and other legumes, and the phytates basically just kind of reduces accessibility by those enzymatic breakdown. The proteases and peptidases don't have as easy access, so we need to break those down to help increase digestibility. Any questions about any of that? Yeah.
Does the fiber bind or does it slow digestion?
Well, there are a number of things that it does, actually. So fiber in the plant food is also, it does slow it slows movement, which could help, right? in some ways because you're kind of moving more slowly through the intestinal tract, but it just tends to reduce accessibility. So, you know, it forms a matrix and structure, and it can interfere with the ability of peptidases to get in and cleave off amino acids. So that's how that tends to work, similarly with phytates. What are the human factors influencing protein requirements? Age, right? We talked about kids having a higher requirement for protein because of growth, right? What about older adults? Do you have anybody read anything about older adults? First of all, let me ask you, what's the RDA for anybody over 18? What's the RDA? I think I heard 0.8, right? 0.8 grams per kilogram. Is it higher for people over, let's see, what is it typically? 51 to 70, and then they've got the higher range people. Do you know? It's not, is it? There's a lot of research going on about protein needs for older adults. And the methods by which we've established requirements are really they're getting a little bit outdated. You know, when we think about, you know, nitrogen balance is a lot of what we talk about. We also, with kids, it's, you know, we gauge it based on growth, right? So we can, and those studies are very difficult to do, if you can imagine. I mean, it's a challenge from an IRB standpoint to reestablish those, so we probably aren't going to do that. But there have a number of studies in older adults showing that there is some potentially anabolic resistance in some people, depending on the level of inflammation that they might have because of chronic disease. They might have some arthritis or some pulmonary disease and that kind of thing. So we're looking at, I mean, that gets us in the realm of, you know, there's this overlap and kind of fuzziness about what's healthy, what's normal aging, and what's kind of pathologic, you know, some of the things that go on. But there are many statements you can find that will talk about how older adults might need more protein, more like 1 gram per kilogram, maybe even higher. It's not set, so the RDA has not been revised, but some interesting things because we lose lean tissue with aging, and that may be better for us, so that may be revised. In your lifetime, it probably will be revised upward. So age influences. What about body size? You know, the larger the person, typically, the higher the protein requirement. Again, how's the RDA expressed? Right, 0.8 gram per kilogram body weight. Now that's been critiqued as well. You know, we talk about this. If we had a way of sizing up people's lean tissue, muscle mass, to maintain that, you know, you're going to have a higher requirement. If you're really a big bodybuilder, you're going to have a higher protein requirement. It's not nearly what people take. I'll just say this, that you know, you don't need to be taking 3 grams of protein per kg per day. In fact, if you think about it, what is anybody ever tried to just eat 0.8 grams per kilogram body weight per day, the RDA? What do you think? Is it more or less than you normally eat? Far less. It's a restriction. It's very uncomfortable to eat 0.8 grams per kilogram per day, just so you know. So most of us eat about 1.5 grams per kilogram per day. Many, especially if you're a meat eater, you're probably eating even more than that. Okay, so you're getting plenty of protein. Even if you were bodybuilding, you don't need to increase your protein from where you've been typically, unless you really are, you know, eating vegetarian and really not eating a lot of animal sources of protein. Physiologic state we talked about, right? Pregnancy, wound healing, bodybuilding, that's kind of the main ones. Growth if you're a child. And then level of energy intake will impact your protein requirements. Why do you think that might be? Anybody have any idea? Why would energy intake influence your requirements? So what if you just, what would happen if you are, well, if you're fasting? What happens when you're fasting? to protein. You're not taking any in. What happens? Muscle breakdown, right? Because you need a certain amount of amino acids. How if you so if you think about that, you're fasting, right? You're not taking in any calories either. So you're going to break down protein, you're going to break down fat or whatever for your energy requirements. You're going to use some of the amino acids probably for energy as well, potentially. But you'll use your fat, your glycogen, and then it'll be gone, and then, whatever. So energy is going to impact your protein requirements. And if you're not taking enough calories from fat or carbohydrate, say you're on the, you're eating protein, but you're not taking in enough calories to meet the needs for your brain. There are certain tissues that are what we say are obligate glucose users. So say you're on the Atkins diet and you're consuming about 20 grams of carbohydrate per day, but you're taking in ad libitum you're eating tons of protein, right? Are you going to use all that protein to support your muscles and all the protein that you need? No, you're going to catabolize it, right? You're going to use it for energy. So you need enough energy coming in as carbohydrate or fat, basically, to spare the protein that you're taking in to use the protein for anabolism. Now your body's not that selective, right? Be nice if it were, but essentially when you're taking in the normal amount of, meeting the minimum requirements for protein, which actually is probably even less than 0.8 grams per kilogram for most of us, right? As long as you're taking that amount of protein in, you got to make sure you're meeting your energy requirement, or you're going to start using that amino acid pool, the carbon, you're going to use that for energy. You're going to oxidize those carbons, right? So protein sparing is what carbohydrate and fat are, essentially. You're providing energy to meet your energy requirement so that you're not using the amino acids to make glucose for your brain, for example, because there's some other tissues in your kidney cortex as well that needs to have glucose. So you got to take in carbohydrate and fat to spare that protein. That's what protein sparing means. And then the RDA for protein differs for different groups of people. I think we've gathered that. It's 0.8 for adults, higher for kids, and you can look those up. I'm not going to ask you. And we said people that have higher muscle are going to have higher protein requirements. That doesn't get reflected in the RDA because we don't specify it for that, but just so that you know, those are the kind of things. Okay, any questions about protein, digestibility, absorption, that kind of thing? Okay, anything jump out at you from the summary? I'm just going to skip on, take it a little. Okay. I think I've got another, I'm not sure if I don't. No exciting animations here. Now we're going to start talking about the amino acid pool and just a little bit more about amino acids, just so we're on the same page. So when we think about an amino acid structure, I talked about this is the alpha carboxyl group, the alpha amino group, and then we got the R group, right? And some of you had already mentioned this, that the R group is really what makes an amino acid unique, right? The R group is the functional group of the amino acid. It's what makes each amino acid unique, and it also is the thing that dictates the amino acid's chemical properties. So the R group is going to determine whether it's a charged amino acid, if it's hydrophobic, or right, we've talked a little bit about this, or maybe it's hydrophilic. They can be small or large, straight, branched, might have sulfur in there. There are a whole lot of them. You remember from your learning all of them, I'm sure, that they're different structures. So, we can categorize amino acids by their R groups and their structure and chemical properties. We can talk about them by essentiality. And then at the end, we're going to talk about by the fate of their carbon skeleton as we begin to move into the next major organ, which I think we'll talk about the liver first. So let's just look at a few structures just to remind you. It's they can be small and neutral, like alanine, that's the smallest one. Glutamate, negatively charged, because it's got this negative charge on the gamma carboxyl group, and then it can be large and positively charged, like arginine has a positive charge. So these are going to be hydrophilic as well, right? But this is when we talk about large being transported faster than small. Those kinds of things are what come into play. I've got another concept that you learned in biochem. When you have an entity like an amino acid that has a no net charge, remember the term? This is another Trivial Pursuit game term. Do you remember, Jonathan? Yay. Zwitter ion. Do you remember that? I like that word. I don't know. I'm going to spell it, though, for some of you. ZWITTERION. Zwitter ion. Means no net charge. So alanine is one of those that we'd call a Zwitter ion. We make more of them when we change pH. We can change glutamate into one. We almost never can do it for arginine. We're going to talk about negative and positive. The other thing about alanine is it's a polar one because there's no charge on it, right? So it's going to be, it's not polar, sorry, non-polar. Golly, forgive me for misspeaking there. Make sure you correct that. Non-polar, which means it's hydrophobic, right? Non-polar is the same as saying hydrophobic. Whereas glutamate and arginine, I mentioned, are hydrophilic or polar. P-O-L-A-R is what I'm saying there. Polar or hydrophilic versus non-polar hydrophobic. Okay. Then, I'm pointing out here, this is the structure of urea. I want you to remember this because you're going to learn the urea cycle. So it's helpful to see arginine as a source of urea. That's where we get urea. The last step of the urea cycle is cleaving off the urea from arginine. So next time we'll get into that. All right, these are cousins. I just like to show the structure of glutamine and glutamate. We're going to talk a lot about glutamine and glutamate because they are very, very important amino acids. Anybody have any idea why glutamine is so important in the body? Anybody have any knowledge, any idea about glutamine at all? I just mentioned that it might be in a formula. Alicia, you remember?
I was going to say good for gut health.
Yeah, it is. Glutamine is good for gut health. And I think I get into this later, so I'll repeat myself probably, and you'll be sick of hearing about glutamine. But glutamine is used by intestinal cells for energy. It's a preferential energy source by the GI tract. So glutamine, when you get out there and you start seeing, wow, what's the deal with glutamine? You're going to know. It supports the intestinal cells because it provides energy to those intestinal cells. Incidentally, immune cells also like glutamine. Immune cells, lymphocytes will take glutamine and use it for energy as a preferential energy source. So gut, GI tract, and immune cells, glutamine is an important thing. While we're writing about it, because I like to write about it the first time you see something. I'm sorry, I'm really cold. My teeth are about to chatter, which might be loud over the microphone. Glutamine, if you think about it, see that purple nitrogen there, right? It's a way of carrying amino nitrogen or ammonia, It's a way of carrying nitrogen around in the body. Very important carrier of nitrogen between cells. It's actually the most abundant amino acid in circulation, going between tissues, carrying nitrogen around. And when it's in the form of this amino acid, that, formerly you can recognize that it's ammonia there, right? It's attached to the glutamine. Now it's part of glutamine. It's not toxic when it's in the form of glutamine, so we can carry it around in the body. It's a great way to get nitrogen from amino nitrogen, which is ammonia, into different tissues. Okay, so that's the number one thing about glutamine. And primary energy source for immune cells and GI tract. Okay, so those are the two big things about glutamine. Glutamate, on the other hand, is a very important intracellular. It's the most important intracellular, meaning inside cells, right? Inside cells, glutamate is the most important amino acid. We're going to know about that after we talk about amino acid, a little bit more about amino acid metabolism. Incidentally, and I'm not showing you the structures, I can't remember, so let me click. Oh no, I didn't, okay. There are some, so I said these are related, right? They're kind of cousins too. There's another 2 amino acids that are very similar in structure. One very similar to glutamine that has 1 less carbon. Anybody remember it? Yeah, asparagine, right? Am I saying that right? Yes. ASN is the 3 asparagine, you know, ASPARAGINE, asparagine. And then what is its cousin that doesn't have the extra nitrogen? Anybody? Cousin directly related to asparagine? I'm hearing it. Say it out loud. Nobody? I'm hearing aspartate, right? Aspartate. So asparagine, 1 less carbon here, has the nitrogen, looks just like glutamine, but it's got 1 less carbon. And aspartate, 1 less carbon, looks just like glutamate. This is the gamma carboxyl group, by the way. So when we up in the R group for glutamate and aspartate, they've got a gamma carboxyl group. All right, R groups influence protein structure and function, right? So hemoglobin, we talked about how it's got this globular structure. It's made out of 2 beta and 2 alpha chains, right? And it's how the amino acid content that forms the primary sequence dictates how the protein folds up to form the tertiary and quaternary structure so it can carry out its functions. So R groups dictate the structure and the structure dictates the function. I mean, hopefully that's obvious to you, but. And this is a no-brainer, right? What mineral is needed to bind oxygen within hemoglobin? So you know the most important thing about hemoglobin is that it binds oxygen and delivers oxygen to the tissues, right? What mineral is needed in hemoglobin? Yay, you can now graduate. Iron, right? Yeah. So we need adequate iron to make adequate hemoglobin so that we can oxygenate the tissues. So when people talk about the major sign of iron deficiency, when you're end of the road really deficient in iron, what's one of the things that is kind of a classic? If somebody's got this symptom, you might say, gee, you know, you might be iron deficient. That's related to this. Fatigue, right? They're dragging. It's all about that. You're not able to deliver oxygen to the tissues. So, all right, great. Oops, too fast. Now let's talk about, so acidic. I showed you a picture of glutamate, right? Had that gamma carboxyl group, had that negative charge on it. Negatively charged amino acids are called acidic. Arginine, remember, was the picture that had the positive R group, and there are some other ones. That would be called a positively charged or basic amino acid. And those will interact with each other, the negative charges and the positive charges, right? Very basic chemistry. Ionic interactions will stabilize the protein structure. So here are some negative ones, right? Again, glutamate is what we call it when it's got a gamma carboxyl group. It loses that at certain points when, you know, we'll talk about that more in a second. It can lose it. If it's got a hydrogen on there, then it's no longer going to have that negative charge, right? And then it becomes what? A Zwitter ion in that form. But as it's, you know, in its normal state, glutamate is going to have that negative charge on the gamma carboxyl group and have that negative charge So, you know, at neutral pH, which is normal physiology, pH 7, it's going to be in this form. Okay? Now, let me think about where I want to talk about that. Yeah, I guess I talk about it here. So this is where we get into biochem a little bit high, a little bit more. Remember the Henderson-Hasselbalch equation? I hated that when I was in school. I really hated the Henderson-Hasselbalch. But it's kind of important because there's the notion of pKa, right? And the pKa of an acid is when you can, well, okay, first of all, Henderson-Hasselbalch, right, was the pH equals the pKa plus the log of base over acid, right? That fun thing that, so the rule that I want you to just keep in mind, in case that you obliterated that from your memory, you know, as I did when I was at your stage, is that protons are going to dissociate or come off. Okay, protons, you know, which are H+, dissociate when the pH is higher than the pKa. I'm going to say that again. The rule is protons, H+, dissociate or come off when pH greater than pKa, when pH is higher than the pKa for a particular group. Okay? So the pKa for this carboxyl group is about pH 4. So take a second and just think about that. So I said at physiologic pH, which is 7, right? That proton is off. Everybody make sense of that? It makes sense to you? So it follows the rule, right? pH 7 is greater than pH 4, the pKa, so that's going to be in that negative form, always at neutral pH. Now if we, let me just, I don't want to get ahead of myself, but let me just, give me a second here. Let me show you another one. Same thing, aspartate. Here's aspartate. I didn't show you asparagine, that would be with the amino group on there, but aspartate and aspartic acid, right? So if you had to guess what pH we're talking about here, you're going to say anything above 4, right? Similarly, or on the other hand, neutral pH is always going to have a positive charge on the basic amino acids. Almost to, yeah, pretty much always, especially arginine, because the pKa for this R group is 12.5. Do we ever get it 12.5 in the body? No, we'd be dead, okay, right? That's really, really a strong base, 12.5. So you're never going to see anything but this in the body, no matter where we are. It's going to be positively charged. And so that rule holds. And I'm not giving them to you, but lysine is another basic one. That's got a very high pKa. Lysine has a pKa of 8.3 on its R group. And then histidine is another one that's basic, but its pKa is around 6. So there's a little bit more play with that one, but I'm not going to ask you those pKa's. But arginine and you know, lysine, you can guarantee they're going to be positively charged basic amino acids at really physiologic pH. And I'm going to stop there because I know you guys are antsy, right? It's 12:35, time to quit. Thanks for your attention. I'll see you on Tuesday, right?